She was told it would not affect the baby. She was told the baby could not feel it yet. She was told the stress was hers to manage and that the womb was a sealed unit, a protective capsule that filtered everything out and delivered only nutrients and oxygen to the developing brain. She believed it because her doctor said it, because the culture reinforced it, because it was easier to believe than the alternative. The alternative being that her anxiety, the one she could not silence no matter how many breathing exercises she tried, was not staying inside her body. It was crossing a barrier that was never designed to be impermeable, reaching a brain that was forming its first neural circuits in real time, and writing instructions that would still be executing decades after she delivered. The baby could feel it, not the worry, not the narrative, not the specific anxious thoughts that kept her up at 3 in the morning. The baby felt the cortisol, the biochemical signature of sustained maternal distress, measured in the parts per million, carried through the blood, across the placenta, and into the amniotic fluid. The baby swallows approximately 20 times per hour. The baby was drinking her anxiety, and the baby's brain was taking notes. The placenta is not a wall, it is a filter, and the filter has tolerances. Under normal conditions, the placental enzyme 11-beta-hydroxysteroid dehydrogenase type 2 performs a critical gatekeeping function, converting active cortisol to inactive cortisone before it reaches the fetal circulation. When the mother experiences an ordinary stress response, a momentary spike that resolves within minutes, the enzyme handles the load efficiently. Almost no active cortisol reaches the baby. But maternal anxiety disorder is not a momentary spike. It is a sustained chronic elevation of cortisol that operates 24 hours a day, 7 days a week, for months. Under chronic load, the enzyme's conversion capacity degrades. Its efficiency drops by as much as 40% in some studies. Active cortisol begins to cross in quantities the fetal brain was never designed to encounter at this developmental stage. And the timing could not be worse. The third trimester is when the fetal hypothalamic pituitary adrenal axis is being calibrated, when the glucocorticoid receptors in the hippocampus and prefrontal cortex are setting their baseline sensitivity. Cortisol is not just a stress hormone, it is a programming signal. And the message it carries is binary, safe or dangerous. Calibrate accordingly. The research documenting genetic and epigenetic transfer of anxiety begins with what is arguably the most uncomfortable data in modern developmental psychology, and understanding it requires both the psychological and the neuroscientific lens operating simultaneously. Begin with the psychology. The concept of intergenerational transmission of attachment patterns has been documented since Mary Maine's adult attachment interview studies in the 1980s. Anxious mothers disproportionately produce anxiously attached infants, and this correlation holds even when controlling for postnatal caregiving quality. This means something is being transmitted before the caregiving even begins. The psychological mechanism is prenatal programming of the stress response threshold. The baby born to a chronically anxious mother arrives with an HPA axis that expects danger. The baby does not know the word anxiety. The baby does not need the word. The baby's stress thermostat has been set to a room temperature that the mother's cortisol described as threatening, and the thermostat does not care whether the description was accurate. It calibrated to the data it received. Now, the neuroscience. The epigenetic pathway that makes this transmission structurally durable involves methylation of the NR3C1 gene, the gene encoding the glucocorticoid receptor. Studies by Michael Meany and colleagues at McGill University demonstrated in animal models that high maternal stress produces increased methylation on the NR3C1 promoter region, reducing the expression of glucocorticoid receptors in the offspring's hippocampus. Now, fewer glucocorticoid receptors means the brain is less efficient at detecting and downregulating its own cortisol production. The system runs hot because it has lost the thermometer. And here is where the data becomes genuinely alarming. These methylation patterns have been confirmed in human neonates. Research by Oberlander and colleagues published in Epigenetics showed significantly increased NR3C1 methylation in cord blood samples from babies born to mothers with third trimester depression and anxiety compared to controls. The babies had not yet taken their first breath, and the stress programming was already measurable at the molecular level. Layer on the neuroimaging data derived from longitudinal cohort studies. Children born to mothers with diagnosed anxiety disorders show measurable structural differences at age 6 to 10, at reduced hippocampal volume, altered amygdala connectivity, thinner prefrontal cortex. These are not subtle statistical tendencies visible only in massive datasets. These are structural brain differences detectable on standard neuroimaging protocols. The anxious mother did not transmit worry, she transmitted architecture. And the architecture she transmitted was designed for a world that her cortisol predicted would be dangerous, regardless of whether the world the child actually entered would confirm or deny that prediction. The prediction was made in the womb, it was written in methylation, and by the time anyone thought to check, the baby had already been built to the specifications the cortisol provided. Now follow the child forward, because the prenatal programming does not express itself immediately in the way most people expect. The baby born to the anxious mother does not arrive screaming. That is one of the most important misconceptions to correct. In many cases, perhaps most cases, the baby arrives quiet, watchful, hyper-alert, scanning the room with a kind of intensity that adults find either impressive or unsettling, depending on their own anxiety levels. The parents describe the baby as sensitive, as observant, as the baby who does not miss anything. The grandparents say the baby is an old soul. The pediatrician says the baby is highly aware. These descriptions are all accurate, and they are also all the visible behavioral surface of a deeper neurological reality that no one in the room is equipped to identify. The baby does not miss anything, because the baby's threat surveillance system, the tonic alertness maintained by locus coeruleus norepinephrine circuits, is operating at a higher baseline firing rate than the average infant. The baby scans more because the scanning system is set to a higher sensitivity threshold. The baby startles more easily because the acoustic startle reflex pathway through the pontine reticular formation is disinhibited by elevated baseline norepinephrine. The baby takes longer to settle after arousal because the downregulation pathway, which depends critically on those underexpressed glucocorticoid receptors in the hippocampus, works more slowly and less efficiently than it does in the average infant. This is the behavioral phenotype of prenatal anxiety transmission. Not panic, not visible distress, not the fussy baby the parenting books warned you about. Vigilance, constant energy expensive, neurologically exhausting vigilance that the baby cannot turn off because the off switch was literally built with reduced sensitivity during a developmental window that is now closed. The parents see a sensitive child. The developmental neuroscientist sees a surveillance system that was calibrated for maximum threat detection during construction and is now executing those exact specifications in a peacetime environment that the system does not recognize as peaceful and may never recognize as peaceful without targeted intervention. Here is where the psychological consequences begin to compound in ways that the prenatal data alone cannot predict. The child with a prenatal anxiety template does not just carry an overactive stress response. The child carries a perceptual filter that changes how emotional information is processed at every level. Cognitive bias research shows that children with elevated trait anxiety demonstrate attentional biases toward threatening stimuli that are measurable by age 3. These children look at angry faces longer. They detect threatening content in ambiguous situations that other children rate as neutral. They interpret uncertain social signals as negative rather than positive. The bias is not learned, it was installed. The perceptual system was calibrated to weight negative information more heavily than positive information, because the prenatal cortisol environment said, the world rewards vigilance, not optimism. Now extend this through the child's social development. The child who perceives threat where none exists generates more defensive behavior. Defensive behavior triggers social rejection. Social rejection confirms the child's model that the world is unsafe. The model generates more defensive behavior. The cycle is self-reinforcing. And it was initiated by cortisol that crossed a placental barrier years before the first social interaction. The anxious mother did not give her child anxiety in the way people typically imagine, as a learned behavior modeled during childhood. She gave her child a perceptual architecture that makes anxiety the statistically most likely outcome, given how the incoming data is being processed. The child is not choosing to be anxious. The child is processing the world through equipment that was pre-tuned for anxiety before the child ever encountered the world. If you are an anxious mother reading this, or if you were the baby born to one, the guilt impulse is going to hit in approximately 3 seconds, and I need to catch it before it lands. Guilt is not the correct response to this research. Guilt requires intent. You did not choose your anxiety. You did not choose to have an HPA axis that runs hot. You did not choose the epigenetic marks on your own NR3C1 gene that may have been placed there by your own mother's cortisol before you were born. The transmission chain did not start with you. It may not have started with her either. The methylation patterns travel through generations like a relay signal, each generation's stress response calibrating the next generation's thermostat based on the data available at the time. You are not the origin. You are a node in a transmission chain that stretches back through every pregnancy that occurred under conditions of fear, scarcity, uncertainty, and survival pressure. The guilt response is not warranted because the transmission was never a choice. But awareness is warranted, because awareness is the mechanism by which the chain can be interrupted. Were you the anxious mother, or were you the baby who showed up already wired for vigilance? Tell me in the comments. The interruption point exists, and it is more robust than the prenatal programming data initially suggests. This is critical, because the most dangerous thing about the epigenetic research is the fatalism it can produce if presented without the plasticity data that accompanies it. Yes, prenatal cortisol exposure programs the HPA axis. Yes, the methylation marks are measurable at birth, but methylation is not destiny. It is a setting, and settings can be changed. The field of behavioral epigenetics has demonstrated repeatedly that postnatal environmental input can modify prenatal epigenetic marks. The mechanism is the same neuroplasticity that allowed the marks to be made in the first place. If the NR3C1 gene can be methylated by cortisol, it can be demethylated by the signals associated with safety, specifically oxytocin, consistent co-regulation, and repeated experiences of stress followed by successful recovery. Megan Gunner's adoption studies at the University of Minnesota showed that children who experienced early adversity, including prenatal stress exposure, demonstrated significant HPA axis normalization when placed in responsive caregiving environments. The thermostat can be recalibrated, not instantly, not through a single intervention, but through the sustained repeated experience of being stressed, being co-regulated by an attuned caregiver, and returning to baseline safely. Each successful regulation cycle writes new data over the old programming. For the anxious mother who is still in the window, the baby is here, the first 2 years are still available, the most powerful intervention is not the elimination of your anxiety. You cannot will cortisol down through positive thinking. The most powerful intervention is co-regulation, being present during the baby's distress, responding with consistent, attuned warmth, allowing the baby's nervous system to borrow your regulation, even when your own regulation is imperfect. The baby's HPA axis is still calibrating. It is still receiving environmental data. Every time you respond to your baby's cry with warmth and presence, you are sending a biochemical counter signal that says this stress resolves and this person makes it resolve. That signal, repeated hundreds and hundreds of times, can overwrite the prenatal prediction. The anxious mother cannot delete the methylation marks she transmitted, but she can generate the conditions under which new marks are written. That is not a consolation. That is a biological mechanism, testable, measurable, and documented. Send this to every anxious mother you know, not to scare her, to arm her with the one thing that actually interrupts the transmission chain. Knowledge that the postnatal environment still has a vote. Come back to the pregnancy. The woman lying awake at 3 in the morning with her hand on her stomach and her mind cycling through every possible catastrophe. She cannot stop the thoughts. She has tried. The breathing exercises help for 6 minutes, and then the cortisol comes back. Because the cortisol is not driven by thoughts. The cortisol is driven by an HPA axis that was calibrated during her own prenatal development, by her own mother's stress, by a transmission chain that may stretch back generations through pregnancies marked by war, by poverty, by domestic violence, by immigration, by famine, by every condition that told a pregnant woman's body the world was dangerous and the baby should be ready. She is not broken. Reishi is a relay in a biological signal chain that has been transmitting survival data from mother to child since before the species had words for what it was doing, and the baby inside her is receiving the latest transmission. The data says the world is dangerous. Calibrate for threat. Set the thermostat high. Build the surveillance system at maximum sensitivity. The baby will arrive built to these specifications, and within weeks, someone will describe the baby as sensitive, as alert, as the one who does not miss anything. And they will be right. The baby does not miss anything because the baby was built not to miss anything. Because missing something in the world the prenatal data described could have been fatal. But here is what the transmission chain could not anticipate. It's the world the baby enters may not be the world the data described. The mother may seek treatment. The environment may stabilize. The caregiving may be warm, responsive, and consistent. And if it is, the baby's still forming neural architecture will receive new data, safety data, regulation data, data that says the threat level has been downgraded and the thermostat can be adjusted. The prenatal prediction is not the final word, it is the first draft. And the first draft can be revised by every attuned interaction, every responsive co-regulation, every moment of safety that the baby experiences in the months and years that follow. The anxious mother did not ruin her baby. She gave her baby a first draft written in the only language the prenatal system speaks, cortisol. And now, with awareness, with treatment, with presence and attunement, she gets to help write the second draft. The one that says you were built for danger, but you landed in safety. And safety, it turns out, is something the brain can learn to believe. Subscribe if this changed how you think about the invisible things mothers carry. The next video examines what happens when a baby who is wired for vigilance meets a world that actually is dangerous. And the answer reveals why some children survive environments that should have destroyed them.